Hi, my name is Jun Tae He, and I'm a graduate student in Biomedical Engineering and Sciences program at Wake Forest University. And I'm working on bioprinting and development of BioInk at Wake Forest Institute for Regenerative Medicine. I'm so honored to present here at the International Conference on Biofabrication 2021 on behalf of our team. And my presentation topic today will be combinations of photoinitiator and UV observer for cell-based DLP bioprinting. 3D bioprinting technologies have been widely used in the field of tissue engineering. Recently, DLP 3D bioprinting has been focused due to its advantages of making a high resolution and complex structures. It has been utilized in various tissue engineering applications, such as bioprinting of tissue scaffold for in vivo implantation or microchannel constructs for in vitro model. DLP BioInk consists of photocross-linkable polymers and cells, but it also requires optimized combination of photoinitiator and UV observer. Photoinitiator plays a critical role by producing free radicals that initiate the polymerization of BioInk when irradiated by UV light. On the other hand, UV observer observes UV light and prevents the scattering of light in the resin to increase the XY resolution in the printed construct. It also prevents overcuring of layers beyond the focal plane, avoiding the undesirable cross-linking inside the hydrogel, and this is helpful when trying to print a hollow channel structure. These photoinitiators and UV observers need to be carefully selected in DLP BioInk since they can critically affect the cell viability and printability in the scaffold. However, there has not been many studies comparing and optimizing those photoinitiator and UV observer combination for cell-based DLP bioprinting. Therefore, the objective of this study was to compare the most used photoinitiator and UV observers in the literatures to find the optimized combination that has the balanced point between DLP printability and cell viability. Initially, candidates of 11 photoinitiators and 5 UV observers were evaluated and screened based on their basic functions. Photoinitiators were first screened based on their ability to effectively cross-link the PECDA solution with minimum UV light intensity and curing time. UV observers, on the other hand, were screened by evaluating their ability to properly block the UV light and create a hollow channel. After initial screening, the cytotoxicity and printability of the selected photoinitiators and UV observer combinations were assessed. As their concentration increases, the higher cytotoxicity and decreased proliferation of C2C12 cells were shown as expected. In the hollow channel printability test, the concentration of UV observer significantly affected the resolution of the printed channels. Based on these results, a combination of 0.2 weight volume percent LAP and 0.5 weight volume percent R1800 showed a feasible balance between the cell cytotoxicity and printability. Then, the optimized photoinitiator and UV observer combination was used to 3D bioprint a scaffold with C2C12 cells. No initial cytotoxic effect was shown, and the cells had an over 90% viability up to day 14. Additionally, the cells were homogeneously distributed in the hydrogel scaffold in both horizontal and vertical directions. In summary, 11 photoinitiators, 5 UV observers, and their combinations were systemically screened and evaluated for cell-based DLP bioprinting applications. The optimized combination of photoinitiator and UV observer showed a great balance between the printability and cell cytotoxicity. This combination may need to be modified depending on the BioInk formulation or DLP printer settings, but it can provide a potential starting condition when developing a novel DLP BioInk. In terms of potential DLP bioprinting, we believe DLP bioprinting has significant advantages when fabricating a tissue and organ mimicking scaffold that requires a complex geometry, such as a heart. It can also be used to develop a higher throughput in vitro model, such as a drug screening platform or an organ on a chip. In conclusion, DLP bioprinting still needs to overcome limitations, but it is definitely a strong tool that has many potential in tissue engineering applications. That is the end of my talk, and thank you so much for listening. I would like to thank every member of our lab who has supported me, and this talk would not have been possible without them. I would also like to acknowledge our funding sources and thank the Biofabrication committee members for giving me this wonderful opportunity. Lastly, I really appreciate the audiences who listened to my talk today. Thank you again, and have a wonderful day.